Bismillahirrahmanirrahim. In this video, we are going to learn about the characteristic, formation, and importance of collagen in the body. Collagens are most abundant protein in the human body, and a comprehensive understanding of the collagen reveals it to be unique triple helical protein family that forms the backbone of animal connective tissue. Collagens elaborate biosynthesis, beginning with gene transcription in the endoplasmic reticulum, involving critical post-translational hydroxylation and glycosylation, and culminate in extracellular assembly into the fibril, is essential for tissue integrity and repair. So, it is the backbone of animal connective tissues. This collagen is secreted by fibroblast. Collagens are important for structural integrity of the organ and tissue, and there are about 28 distinct collagen molecules numbered from 1 to 28. They are triple helices of homo or heterotrimers of alpha chains, and the amino acid motif that are making these collagens are repeating glycine XY, and these XY are commonly proline and hydroxyproline. Post-translational requirements are necessary for the structural integrity of these collagen, and this is dependent on ascorbic acid or vitamin C dependent hydroxylation of proline and lysine. There is tissue-specific covalent cross-links via lysine, hydroxylysine, and histidine. The types of collagen, the major type of the collagen are two. One is the fibrillar, that forming stiff, uninterrupted triple helices, and examples are type 1, 2, 3, 5, and 11, while another form is non-fibrillar type of the collagen. It contains flexible, interrupting, non-collagenous domains. Key functions of these collagen molecules are structural framework, cell growth, migration, differentiation, and matrix morphogenesis. It interacts with integrin and discoidin domain receptors. So this is its interaction with the receptor. Whenever it is decreased, reduced, delayed, or abnormal, it will lead into scurvy or non-healing wound, while increase in this collagen, increased deposition of the collagen will result in fibrosis as seen in scleroderma, keloid, and chronic organ damage. As I just mentioned, there are about 28 types of these collagen, and out of these 28, the type 1 of the collagen is about 90% of the body collagen, and it is present in skin, bone, tendons, and provide a tensile strength, while type 3 is present in skin, vessel, early wound repair, and in the wound it is later replaced by type 1 of the collagen. Type 4 collagen is found under the basement membrane, and it support the epithelial or other structures that are resting on the basement membrane, and also its function is to filtrate. Type 7 and 17 are part of hemidesmosome, and they are responsible for anchoring and provide a stability to the skin. Type 6 of the collagen is pericellular matrix. It support muscle fiber and interact with elastin. The basic structure of the collagen is a triple helix of 3 alpha chains. These can be homotypic or heterotypic. Alpha chains may be of a single type or there may be of a different type. The types of trimers are homotrimer that are present in collagen 12 and collagen 18, or heterotrimer, and these heterotrimer are present in a specific site. There are specific combination that are present in a specific site. Then there is a special case in is collagen 4. It assembles from 6 alpha chain into at least 3 heterotrimeric isoforms. Key motifs for this collagen are, as we discussed earlier, these are glycine XY, and the glycine is present on every third residue, and it is important for triple helix stability. The amino acid involved for its formation are glycine, proline, hydroxyproline, lysine, hydroxylysine, and histidine. And there is covalent cross-link formed intra and intermolecularly that are tissue specific. If there is mutation in glycine XY sequence, the helix will be unstable and it is prone to degradation by proteases. After the gene transcription, post-translational modification in collagen synthesis are very important. So there is first one is the chain selection. Based on the chain recognition sequence, prolyl hydroxylation aids correct alignment. After that, hydroxylation. This hydroxylation is ascorbic acid or vitamin C dependent of proline and lysine. Then there is formation of helix from aligned alpha chains. These are packaged into COP2 vesicle for transport from endoplasmic reticulum to the Golgi apparatus, and glycosylation occur in Golgi apparatus. The sixth step is cleavage of the terminal peptide. So non-collagenous terminal domains removed before fibril formation, and molecular alignment is done and microfibril formed. Final stabilization via lysyl oxidase, that is a copper dependent enzyme. Some of the byproducts of this collagen that are made during the synthesis and remodeling are used as biomarker for the detection of ongoing fibrosis and development of osteoporosis. If these collagen gene is mutated, it may result in some specific condition. If there is damage or genetic mutation, it will lead to the osteogenesis imperfecta, or if the cartilage collagen is deficient or defective, it will result in achondrogenesis or hypochondrogenesis, chondroplasias, or epiphyseal dysplasias. The defective collagen of skin, joint, and vessel will result in the manifestation of Ehlers-Danlos syndrome. This table is summarizing the salient features of fibrillar type of the collagen. The fibrillar type of the collagen are type 1, 2, 3, 5 and 11. Type 1 of the collagen is most abundantly present in the skin, bone and tendon, and it is present as uninterrupted triple helix. There is slow turnover and it forms strong fibrils, and if there is any mutation there will be increased fractures and extensible ligaments, and there will be easy bruising. Type 2 of the collagen is present in the cartilage and it forms continuous fibrils nucleated by type 11. These are essential for cartilage structure. Type 3 of the collagen is present in skin and blood vessel, and it is associated with type 1 for assembly of the fibril, and it supports structural integrity. Type 5 collagen is widespread. It is present in the skin and placenta. It nucleates type 1 fibril also and control fibril diameter. It regulates type 1 fibril formation. Type 11 of the collagen is present in the cartilage and it nucleates type 2 fibril, and it is key for the cartilage matrix formation. The common properties of this fibrillar collagen are quarter scattered arrangement of the triple helices. These are resistance to proteinase digestion except matrix metalloproteinases. If there is mutation, there will be pathology, and these pathologies range from mild to lethal types. There will be a structural defect that lead to connective tissue disorders. This diagram illustrates the fibril forming type of the collagen and their self-association pattern, and they are important to understand their structural role in the connective tissue. Here, the types of the fibril form are type 1, 2, 3, 5 and 11, and they are arranged in the skin, bone, cartilage and blood vessel. And here you can see the macromolecule. It is self-associated in staggered fashion or staggered array. Each arrow over here represent a collagen triple helix, and the staggered alignment, the overlapping ends, the staggered alignment, overlapping ends lead to characteristic banded appearance of the collagen fibril. Over here, as you can see, the banded appearance of the collagen fibril under electron microscope. This scattered overlap increases the tensile strength and forms the backbone of extracellular matrix architecture. The electron microscopy, over here you can see the type 1 and type 2 fibrils are there, and type 1 and 2 fibrils are showing periodic bending that is seen in electron microscopy as light and dark repeating bands due to the staggered arrangement. Type 3 fibrils, as is evident over here, these type 3 fibrils appear as a uniform line with periodic nodular structure that indicates structural difference in packing or glycosylation. The non-fibrillar type of the collagen are many. They are 9, 12, 6, 7, 17, 4, 8, 10, 15, and 18. The 9 and 12 are mixed globular and triple helicular, and they are small collagens. They bind fibrillar collagen, especially type 1, and they modulate the packing of the fiber. So their function or clinical relevance is structural modulation. Type 6 of the non-fibrillar collagen is beaded filament that is found near the cell. It encircles collagen 1 and 2 and connect muscle to the basement membrane. Mutation in this type 6 of the collagen result in myopathies and muscular dystrophy. Type 7 of the collagen are anchoring fibrils. They link hemidesmosomes to the basement membrane and stroma, and their mutation result in dystrophic epidermolysis bullosa. Type 17 of the collagen is a transmembrane protein that is found in hemidesmosomes and attaches epidermal cell to the basement membrane. Its mutation really leads to junctional epidermolysis bullosa. Type 4 of the non-fibrillar collagen is a network forming basement membrane, and it has got flexible chicken wire network. Mutation of this type 4 collagen results in Alport syndrome, and fragments inhibit angiogenesis. Type 8 and 10 type of the collagen are hexagonal networks. They have got a structural role in specialized tissue, and their clinical relevance is structural matrix integrity. Type 15 and 18 are present at the basement membrane stromal interface, and their fragments or matrikines, they inhibit angiogenesis and tumor growth like type 4 fragments. So their clinical relevance is in anti-tumor, anti-angiogenic property. So the non-fibrillar collagen has got some key features. One is that it contains interrupted triple helices with globular domain that imparts it flexibility. It forms network, anchors, or beaded filament rather than long fibrils. It supports basement membrane, modulate fibrillar collagen behavior. It generates matrikines. These are biactive fragments with their role in repair, angiogenesis, and cancer. So here a visual overview of the non-fibril forming collagen is given, and you can see that this non-fibrillar form of the collagen differs by the from the classical fibrillar collagen by having interrupted triple helices domain. Interrupted triple helical domains are there, and their function is beyond the structural support. So the types of the non-fibrillar collagen are type 6, 9, that is specific to the cartilage, and it is also a proteoglycan. Type 12, type 15 and 18, also 18 proteoglycans, 15 and 18, and These collagens are essential for connecting, anchoring, and modifying the extracellular matrix, often interacting with fibrillar collagen. The type 4, the type 6 of the collagen, it formed dimers and then it formed tetramer, assembling this into a beaded filament. These filaments provide flexible connection in extracellular matrix, especially in muscle and connective tissue. Type 9, it is associated with type 2 fibril that are present in the cartilage, and they are attached with glycosaminoglycans side chains, reflecting its role as a proteoglycan which adds compressive strength and hydration. Type 12 act like a bridging molecule connecting the collagen fibril, bridging molecule connecting the collagen fibril to the surrounding extracellular matrix component. So this radiating in star-like shape, and this indicates multiple interaction points for this type 12 collagen. Type 15 and 18 also shown with glycosaminoglycan side chains, and they are also having the protoglycan nature. They modulate cell signaling, basement membrane stability, and angiogenesis, especially type 18. So here the aggregate form is showing the association of the fibril. These collagen interact with classic fibril forming collagen. Type 9 interact with the type 2 fiber. Type 12 interact with type 1 fibrils. These associations help to stabilize the extracellular network, particularly in cartilage, skin, and connective tissue. So these non-fibrillar collagens are regulators and connectors, essential for extracellular matrix, architecture, biochemical integrity, and signaling. So they are just not the structural support, they have got multiple cellular function that are important in homeostasis. Here the network forming types of collagen are presented, and these differ from the fibrillar and non-fibrillar types of the collagen by assembling into mesh-like network. They assemble into a mesh-like network like this rather than the linear fibrillar pattern. So type 4, 8 and 10 are the types of the collagens that are forming networks. So type 4 collagen is found in the basal lamina or basement membrane of the epithelial tissue. So this type 4 of the collagen is found in the basal lamina, and it is part of the basement membrane of epithelial tissue. Type 8 is present in vascular endothelium, and type 10 is found in hypertrophic cartilage, especially during the bone growth and the development of the bone. So, so far as the structural assembly is concerned, it is composed of long triple helical domain with 7s domain at the N terminus and NC1 domain at the C terminus, carbon terminus. These domains allow lateral and end-to-end cross links forming sheet-like networks. So here you can see that this type 4 is forming a 2D meshwork essential for structural scaffold of the basement membrane supporting epithelial cell and filtering substances in tissue like the kidney glomerulus. Type 8 collagen network is in 3D, and here these are forming hexagonal lattice-like structures. These are contributing to the vascular and ocular tissue integrity. Its unique crystalline network support mechanical stability and specialized function like light transmission or endothelial support. The network forming collagen do not provide any tensile strength like fibrillar type that gives, but they are crucial for filtration, compartmentalization, and cellular anchoring within the extracellular matrix. Type 17 and type 7 collagen, they serve as anchoring collagen that is depicted in this illustration. So here these are essential for the role of epithelial adhesion to the underlying connective tissue with the basement membrane and hemidesmosomes. So here these type 7 anchoring collagen, they form long anchoring fibrils, and these fibrils are diameter that loop from the basement membrane into the dermis. These diameters, they loop from the basement membrane into the dermis, securing epithelial cell, while the collagen 17, that is also termed as bullous pemphigoid 180 or bullous pemphigoid 1 antigen 2, is a transmembrane collagen that is found in hemidesmosome, and it spans the plasma membrane from basal keratinocytes linking the intracellular cytoskeleton to the extracellular matrix. And here the aggregate form is reflecting the anchoring mechanism. So type 17 collagen anchor within the hemidesmosome, this area, and type 7 collagen fibrils, they extend from the hemidesmosome uh, from the basement membrane into the papillary dermis, looping into the anchoring plaque that is present in the stroma. This forms a strong mechanical link between the epithelial cells and underlying tissue. So defects in these type 7 collagen will these type 7 collagen anchoring fibril that will lead to dystrophic epidermolysis bullosa and autoantibodies to these type 17 and type 17 collagen fibers will lead to development of bullous pemphigoid. So type 7 is anchoring fibril, while type 17 is transmembrane link in the hemidesmosome. Together, they stabilize skin structure and prevent blistering from mechanical causes or mechanical stress. This is a transmission electron micrograph that is showing the stromal layer of the human cornea. So here you can see that here distinct lamellae or layers of the uniformly arranged collagen fibrils are present, and these layers are sharply defined and arranged like plywood pattern. Sharply defined, each lamella oriented nearly perpendicular to the adjacent one. This magnified colored schematic zone is one of the lamella that is showing the parallel collagen fibrils. These fibrils are highly uniform in diameter and spacing. So this is a critical feature of corneal collagen. Due to this precise orthogonal arrangement, there is minimization of the light scatter that ensure corneal transparency and optimal refraction for clear vision. The collagen type here is type 1 along with type 5 to regulate the fibril diameter. Disruption of this organized structure due to trauma, keratoconus, or corneal edema can impair vision by causing the scattering of the light and haziness. Now we will briefly discuss the tissue-specific collagen organization and their function. The cornea has got type 1 and type 5 of the collagen, and there are 10 to 20 orthogonally stacked uniform sized fibril that are present in transparent layer, and their main functional outcome is transparency and refraction of the light. If there is any injury and infection, that will lead to disorganized opaque scar. As a result, there will be loss of the vision. The skin contains type 1 and type 3 type of the collagen, and here it is arranged in loose basket weave network. The wrinkle lines in the body follow the orientation of the collagen. Their specific function is flexibility and giving tensile strength. The clinical relevance of this collagen that is present over here, that it provides wrinkle line. These wrinkle line guide surgical incision for better healing. Skin type 1 disorganized presence is thicker, it is poorly woven fiber and disorderly patterned, and it gives weaker mechanical strength and aesthetic distortion. And clinical relevance is poor healing, less tensile strength, and visible scarring. Type 1 is also present in tendon and ligaments. Here the parallel array of thick collagen fibrils are present, and there is presence of high tensile strength that is present in one direction, and it is important for joint stability and mobility. The collagen was given name of scleroprotein that emphasizes their white color and stiff or hard property. It is oriented in such a fashion that is responsible for its function. As happens in the cornea, uh, they are arranged in such a fashion that they impart transparency to this organ, while in the skin they give flexibility because they are arranged in basket weave appearance, and in tendon they are parallel to each other and they give strength in this area. The incision that made parallel to the wrinkle line or wrinkle line, they heal better with less scarring. If there is disruption in the collagen organization, as occurs after trauma, it may lead to functional impairment like corneal opacity or disfiguring scars. So here is the comparative table for the fibrillar and non-fibrillar type of the collagen. The examples for fibrillar collagen are type 1, 2, 3, 5 and 11, while type 6, 7, 9, 12, 17 and 27 etc. are non-fibrillar type of collagen. The fibrillar type are long, rigid triple helices without any interruption, while the non-fibrillar type are mix of triple helices and globular non-helical types of domain. Fibril formation, the fibrillar type form long continuous fibril that are arranged in bundles, while the non-fibrillar collagen, they do not form any classic fibril. The fibrillar type is arranged in quarter staggered cross-linked packing, while the non-fibrillar are associated with fibril or membrane not forming any large fibril. The fibrillar type of collagen gives tensile strength and structural support in skin, bone, cartilage, etc., while the non-fibrillar type provide flexibility, connect tissues, anchor cells, and form specialized networks. The fibrillar type is present in skin, bone, tendon, cartilage, and ligament, while the non-fibrillar type is the part of basement membrane, muscle connective tissue, and hemidesmosomes. The fibrillar type is slowly turnover, highly resistant to degradation, while the non-fibrillar is more dynamic and can undergo remodeling. The associated diseases with fibrillar type are osteogenesis imperfecta and Ehlers-Danlos syndrome, while non-fibrillar type of collagen are associated with epidermolysis bullosa, Alport syndrome, and muscular dystrophies. The unique feature of fibrillar is that type 1 is most abundant and type 5 and 11 nucleate the fibril formation, while type 6 is beaded in appearance, beaded filaments. Type 7 is forming anchoring fibrils, and type 17 has got a transmembrane role. The biological activity of fibrillar type is structural primarily, while the non-fibrillar collagen, it regulates the cell adhesion, signaling, and angiogenesis via fragment production that are also termed as matrikines. Here is a summary video for the collagen.
Collagen. You've heard the buzz, but behind the hype lies one of the most vital proteins in the human body. Let's break down its structure, function, and clinical significance. Collagen is a structural protein synthesized primarily by fibroblasts. Its formation begins with transcription of collagen genes producing pre-procollagen chains. These undergo hydroxylation, vitamin C dependent, followed by glycosylation, triple helix formation, and secretion. Extracellular enzymes like lysyl oxidase help create strong cross-linked fibrils. There are over 28 types of collagen, but 4 dominate clinical relevance. Type 1, found in skin, bone, tendons. Type 2, cartilage. Type 3, reticular fibers, organs, vessels. Type 4, basement membranes. Each type is tailored to mechanical demands of specific tissues. Collagen provides tensile strength, tissue architecture, and scaffolding for cell migration and differentiation. It's indispensable in wound healing, bone mineralization, and joint integrity. Collagen synthesis declines with age. UV exposure and smoking accelerate its breakdown. This manifests as wrinkles, joint pain, and delayed wound repair, hallmarks of tissue aging. In aesthetic medicine, collagen is both the target and the tool. Procedures like microneedling, laser therapy, and dermal fillers aim to stimulate or supplement collagen. Oral collagen peptides are marketed for skin elasticity, though evidence remains mixed. For medical professionals, collagen isn't just a cosmetic buzzword, it's a critical player in both health and healing. Understand it well, and you'll see it everywhere, from dermis to disc, lab bench to clinic.
Thank you for watching this video. If you have any query, please write in the comment box.